Good afternoon, Professor Thelma. This is Sachin Chakravarti here from RIS.
Yeah, good afternoon. Thank you very much. Yeah, thank you very
much. Thanks for agreeing to chair the session.
It's a pleasure.
Thank you. Hi, hi, Rudy. How are you?
I'm good. Thank you really for this opportunity.
Bangalore. Yeah, no, you're most welcome. Hi, Dr. Devajyoti.
Hi, hello.
Good afternoon and welcome to this session. Very good.
Thanks for the invitation. Yes.
No, it's a great pleasure. I had spoken to Dr. Anurag and he was so much appreciative of the great work that you are doing. And of course, you are very widely recognized in this area as a technical expert. So that's a great acknowledgement. And for us, it's a great pleasure to have you with us.
Thank you. Looking forward. Thanks for organizing this.
Great. No, it's a great. I'm just waiting. I think Dr. Reddy has also already logged in. So once he switches on, yes, welcome, Dr. Reddy.
Thank you, sir. Thank you, Secretary.
Thank you. So we are dot at 4 o'clock, and I think the public telecast has already started, and quite a few people have joined in. So Dr. Thelma, if you permit now, should we start? Are you all set?
Yes, please. And I just wanted to ask, would you prefer that we keep our videos off or?
I think in the beginning we can keep it on so that our people can take a picture of all those who are participating. And once we are done, Devajyoti, can you also please switch on your video? So we will just take a picture and then we can get it going. In the next 1 minute or so, they would click in, and I would start because people have already started pouring in. So welcome, friends. Warm welcome to this STIP lecture series, Science, Technology, Innovation Initiative of RIS, where we have been discussing issues that are related to science, technology, and innovation, its impact on society, its contribution for our national interest, and also the reminders that are needed in terms of the governance of technology. RIS through STIP program has been discussing several of these issues. As you are already aware, RIS is a policy research institute focusing on the economic, the ethics, and of course, the broad dimension of trade, finance, and technology linkages. It's a think tank supported and funded by Government of India through the Ministry of External Affairs. We try to connect our work with the external sector and try to see what a role the think tank community can play, as it is called, on track 1.5. We try to see the global linkages of technology, how technology may strengthen our international cooperation, international partnership, regional cooperation, and from that point of view, how it determines the trade interests and the interests that are related to economic development of countries. Friends, this season, when we get Nobel Prizes for various spheres and fields, we are reminded of the new frontiers of technology, but we are also reminded of the limitations of technology, the governance part of technology. And this year, with the Nobel Prize going to CRISPR, I think not only the economic development contours are right in front of us, but also the grim reminder of ethics part of it, because what happened in China last year, none of us have forgotten that. So the ethics dimension of it, the moral compass that is needed along with technology, and for countries like India and other developing countries, particularly in Africa, CRISPR offers immense opportunities when it comes to health or agriculture. And these dimensions of CRISPR are extremely important when we think of the delivery strategies, when we think of the technology and the computational analysis, or when we think of development and application of CRISPR. So that way, the area, the landscape is huge and so wide that we require some sort of understanding as to what exactly the CRISPR is. And we thought that some of the best brains in India, those who have been contributing in enhancing India's competence in that area, and we thought of none other than Professor BK Telma to chair the session, and I'm so glad we have a very accomplished a person in form of Professor Telma to chair this session. She has been member of all the three science academies of India. She has been member of Prime Minister's Scientific Advisory Council. She has been part of the National Science and Engineering Research Board and Human Genome Medicine Genomics Task Force. She has also been taking a lot of interest in genomics, and we would hear more from her. Has also been immensely contributing at the South. Part of the Department of Genetics at University of Delhi. She is accompanied by two of our lead speakers from technology sector, Dr. Devajyoti Chakravarti and Dr. MK Reddy. Both of them would be introduced to you by Professor Telma. And we have Dr. Roli Mathur from Indian Council of Medical Research, who would bring the dimension would be important in terms of how we look at the governance part of the technology. What I liked most when the scientists who got the Nobel Prize were being interviewed, and I was very much impressed to see the interviews. I think it has happened for the first time in the history of Nobel Prize that one prize has gone only to the women scientists, and the interview that appeared in I think it was in BBC when one of them who very forcefully actually Aminal very forcefully Aminal said that I do not like to be called women scientist because there are no men scientists. So a scientist is a scientist. No point in creating distinction. But when she was asked what and her that could go to a foundation that her husband at her university for supporting women scientists. So I think all of us realize the challenges. All of us the sort of underprivileged sections also realize where action is needed, and from that point of view, I think the idea of CRISPR as a technology for agriculture interventions, CRISPR as a technology for access to medicine for all, probably are some of the important issues that that we need to focus on. It would also have immense ability to wipe out specific species, and that's what frightens some of us because that's where ethics comes in. The idea that mosquitoes can be wiped out if you use CRISPR technology. We already know, as I mentioned in the beginning, the editing of human genome and the birth of the 3 babies for sickle cell anemia, I think are some of the interventions that have called for the global understanding and convergence of opinion on ethical dimensions of it. And that I think is important. I'm sure the panel led by Professor Telma would certainly reflect on what kind of technological strengths are needed. I'm extremely thankful to my team at RIS led by Professor Ravi Srinivas, who could put this panel together and could bring out the salient features that are related to science, technology, innovation. Friends, as you know, at RIS, we are very closely following the implementation of Sustainable Development Goals. And with the support of the Principal Scientific Advisor, we are running a program on how science, technology, innovation can help us achieve Sustainable Development Goals. And with Ministry of Science and Technology, Department of Science and Technology, DST, we are running a major program on science diplomacy, which is led by Dr. Bhaskar Balakrishnan. So both of them, I'm extremely thankful to Ravi and Dr. Balakrishnan for their support and bringing this event together. So without further ado, I would very much like to request Professor Thelma for her inaugural remarks and for taking the process forward. So you have the floor, Professor Thelma. You are on mute.
No, I'm good.
Thank you.
Yeah.
So the floor is yours.
Yeah, thank you very much. Okay, good evening, everyone. We've gathered this eve to hear a little about CRISPR, which is yet another technology which has emerged big and powerful in less than a decade and attracted the attention of so many researchers in the life science area, be it genome scientists or clinicians or the industry even. The Nobel Prize 2020, please may I have an opportunity to share my screen?
Yes, you have. Please go ahead.
Yeah. Okay. Is that visible?
Not really. I think you have started, but there's nothing coming on the screen.
No, it says you're sharing your screen. Share. So it's sharing my screen according to what I see.
Yeah, it's coming up. Yeah.
It's coming up, right?
Yeah.
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 Please go
Is that
ahead.
right? Yeah. Okay. All right. So, the Nobel Prize 2020, which in chemistry was awarded to two bright and beautiful women, speaks for itself and the power of the technology. Therefore, it's only timely that Professor Chakravarti and his team at RIS have so thoughtfully organized this meeting, including the two essential beneficiaries, which is agriculture and health. And even more importantly, in my opinion, the oversight mechanism of this double-edged sword. I wish to congratulate the team to start with for this first step, and I sincerely hope that this marks the beginning of many more scientific discussions and public debates on the way forward with this very powerful technology. And I'm very thankful to Professor Chakravarti for giving me this opportunity to present the main findings of one such deep deliberation as a part of the International Commission on the germline genome editing for clinical use. I emphasize here, we're talking about the germline or the heritable genome editing and not the somatic cell genome editing. And if you look at history, you can see that over the last 5 decades, we have had major technologies which have come in, and each one has been, very, very important, be it recombinant DNA followed by transgenics, or was it gene therapy or stem cell therapy, and more recently, the next generation sequencing technologies and the present CRISPR. Every one of them has addressed a lot of issues. And in the context, even if I restricted myself, because Professor Chakravarti already gave you a broad picture of all the uses of CRISPR. So even if I kind of restricted myself and talked about only the human genetic disease burden, we talk about approximately 8% of all human genetic diseases are caused by mutations in single genes, and which includes the classical examples which everybody knows, sickle cell anemia, thalassemia, and the like. And over 60% are polygenic disorders, and which include be it hypertension or diabetes or asthma, cardiovascular diseases, etc., where it's not one gene but several genes which are involved. But if you look at the medical, the dogma in medical biology, it essentially is diagnosis, prevention, cure, and treatment in that order. So where does CRISPR fit in this scheme of events? Definitely it has one leg in prevention and another one in cure or treatment depending on the context. So what I want to kind of emphasize here is that while we are looking at the diseases and while we are looking for prevention, we have several methods which are available if you know that there is a genetic disease which is running in the family, or there is a population which is prone to a disease. You can definitely look at that. And if you know the genetics of that, you can have enough genetic diagnostic tools which will help prevent. But in today's presentation, I would actually touch upon a condition where we would like to prevent the mutation which is causing the disease being passed on from the parent to the progeny. So that's really the context that we're talking about. About cure or treatment, you will hear more from Dr. Devajyoti, where he will probably speak about his own example and tell us. And so if we talk about genome editing, and if we look at what really are the possibilities, to cut the long story short, you know that the technique is so powerful that we can have a lot of things which can be done. We heard about the rogue scientist in 2018 who presented in the International Genome Editing Summit the CRISPR babies, which shook the scientific community. And this actually highlighted the absence of the societal acceptance of this potential application. And also it told us that we do not have necessary scientific evidence which is needed to demonstrate the safety of this procedure for clinical use. And it's extremely important in that context that we really look at what is doable, what should be done, what should not be done. And therefore, without going into too much of the technological details, all that I want to emphasize here is that it was at this point in time when you knew that the technology was just emerging, and yet there were scientists who were already taking it to clinical use without proper governance or without proper oversight mechanisms, we could end up with disaster. And that was really the beginning of the need to realize that there is, there has to be a systematic evaluation of the science as it stands today, and also what we could look for possible regulatory mechanisms. So in the next few slides, I will quickly share what we as a group of commissioners with about 26 academies involved, and we were a total of 18 participants from 10 countries who put together this report after a lot of serious deliberations over a period of 1 year. So basically this commission was assessing very critically all the science which is there, and we knew that there is an urgent need to develop a translational pathway for heritable human genome editing. So I emphasize here again that we're not talking about editing the genome for a particular disease like sickle cell anemia or thalassemia, which is done by multiple ways, but let's assume it's only done for that individual. Whatever the edit is there to cure or treat is restricted to that individual and does not get passed on to the progeny. Whereas here we are talking about and taking note of what is important if we are talking about heritable human genome editing, which is what was actually raised, or which was actually kind of, became very evident with the CRISPR babies news, which came in 2018. So there were evidence gathering workshops and a public call for evidence also in the first meeting, and every continent except Antarctica represented between commissioners, speakers, and reviewers. And the commission's task was essentially to develop a framework for a responsible clinical translational pathway for initial uses of heritable human genome editing, and in the event that society decides to permit them. So right there, the societal consensus comes in a big way. And to India, this is not new in terms of a debate where public is involved, because our GM crops are an excellent example where there's been so much debate between scientists and between the public and so on. So it's a very similar situation, but probably far more serious when it comes to talking about taking human evolution in your own hands or playing God. So considering technical, scientific, and other regulatory requirements, especially for clinical use, and societal and ethical issues, which are all, kind of interlinked, were really the task of this commission to look at that carefully. And we did not, of course, look at the societal and ethical issues in a very big way because we had WHO, which was going more into the governance and these issues. The key messages up front then are that as of today, with the existing technology, with the existing evidence for the safety of the procedure, we say that no clinical use of SHGE should be considered until it has been clearly established that it is efficient and it is also precise while making changes and it is safe. However, the Commission made sure that we don't talk about putting an end to this kind of research. We say further research is absolutely necessary because technological development, especially for the medical sciences or for treatment of diseases, is very important. Second key message was before any country decides to approve the use of HHGE, which I'll use as a short form, there should be national and international mechanisms to ensure that the preclinical requirements have been met for initial responsible use. And any clinical use of this technology should proceed cautiously. And to start with, it should be restricted to a limited set of circumstances as per the criteria which are set in the report. So the emphasis was on safety and efficacy of the technologies. And of course, it would depend on the science of the genetic disease, the mode of inheritance, the kind of mutations, the number of genes which are involved, etc. And we, at the end of the meeting, deliberations, realized that at the moment, the criteria for safe and effective HHGE have not yet been met. We might have precise on-target changes, but there are also off-target changes, which are even difficult to assess as of today. So given that, what's the translational pathway that we put forth for initial use? We say that there has to be sufficient preclinical research in cell and animal models to develop and validate the editing methodology. There has to be sufficient evidence with work from human embryos to show the reliability of on-target and precise on-target changes. And there should be no off-target changes. And one of the big issues with this technology, which gets mosaic patterns also, should not be there during this process. Clinical evaluation of embryo to be used in a pregnancy has to be verified to ensure that there is the intended edit and not any of the unintended changes, even if you were to take a part of the biopsied cells. Comparable developmental milestones also needs to be looked at, and protocols, of course, for seeking informed consent and long-term follow-up, etc., are essential. So categorizing the applications by HHGE, it is very clear that you needed to have a phenotype which is going to be influenced, and that needed to be edited. There had to be a very strong correlation between mutation and the phenotype that we are interested in, and what were the options which were available to prospective parents who were at a risk of passing on the genetic disease without the child getting affected. So we put together 6 categories, and the category A and B are really what we believe, if at all the initial users, it should be restricted to that. And we say serious monogenic disease in which all children would inherit the disease. And the second would be serious disease in which some children would inherit the disease. And then, of course, there are other categories like monogenic conditions, but with less serious impacts, doesn't call for such an extensive and a very risky procedure like any genome editing. Polygenic diseases, we are not yet there because it's too many genes which need to be then edited. And then category E is not really for a disease, but for a trait improvement. Let's say intelligence or height or anything that you think is important and you want to manipulate that. And finally, you might have a set of conditions which cause infertility. And this is different because here we are not talking about passing on the benefit to the baby, but then we are talking about the couple themselves getting the benefit. Just to emphasize to the audience what we mean by category A and B, I just have put this slide just to tell you that if we assume that all the alleles which are marked in red are the disease-causing ones, the left panel shows that you have a parent who's affected and he has both copies of his gene which are mutated. All children will be affected. No opportunity for them to have a child who's going to be unaffected. Similarly, when you talk about autosomal recessive, and if you have both parents affected, no chance of the children having a normal situation. And therefore, we consider these as the most serious and under category A. Category B, you can have parents who are carriers. And of course, I've got a mistake there. It's the capital A, the parent should also be affected. And you have not a homozygous unlike the top, and you have a heterozygous where 75% will be affected. And you have another situation where one parent is affected and the other parent is only a carrier where we have 50% affected. So after much deliberations, we said category A and top of category B is what really needs to be taken forward. So the 4 criteria that must be met in all this is serious monogenic disease, and it has to be changed to a common variant, which is not going to be causing a disease. No unaffected embryos are subject to editing and transfer, and it has to be only those couples who have absolutely no or very poor options for having the genetically related child, like what I showed you in some examples in the previous one. So how common are these? Of course, category A are going to be far and few, and category B will definitely have a few of them, comparatively more of them, but it will depend on the country, it depends on the population, and especially these will be in populations, like say, for example, even in India, you will have a higher rate of some of these diseases because of the higher rate of consanguinity. So be it thalassemia, or maybe in some tribal populations, you have sickle cell anemia and so on, where you have a greater chance of having both couples affected. And what should be the governance mechanism? National mechanism is decisions ultimately about whether a country allows HHG or not. It has to be made by that specific country, which means there has to be a lot of debate, but it also should look at mechanisms and competent regulatory bodies which are in place before one can actually go forward. No clinical use of HHG should occur in that country if they're not able to meet these conditions. And it has to be a case-by-case evaluation and transparency, and it has to be also in the light of what the international assessment is. In terms of governance for arrangement of HHG international, so one of the recommendations were that there should be an ongoing evaluation of science and technology in all these efforts. And international scientific advisory panel, which will look at these aspects, was suggested. And then there has to be debate whether it could be appropriate to cross this threshold for a new class of users, depending on the ethical and societal concerns raised. And finally, of course, a mechanism for whistleblowing, which is very essential. So we don't specify in our report what that mechanism should be, but we're almost sure that WHO, which is kind of working in parallel to come with the governance mechanisms, will have it. And taking forward, finally, the Commission's work. Of course, this is a very good example where RIS is put together. More of such meetings and conferences should be there, where people appreciate the benefit of the technology as well as are completely aware of the limitations and the risks and the harm that it imposes or that it brings along. And we believe that WHO, as I said, we will be looking at the global standards for governance and oversight of this powerful technology. And of course, revisiting the conclusions and recommendations at the 3rd International Summit on Human Genome Editing is something which they believe is going to be there. And with that, I close, and I want to tell you that this report is available at the site. And of course, just very recently, we have had over 30 scientists who have commented or who have reviewed and given their reactions to this report. So anybody who's interested in knowing more could please look at this, and you can see the debate which is on. And I want to just one of them actually said, you're building an expensive bridge to a remote island before we know if you're ready to use it. Now, all science has to be progressive. You cannot just stop science with fear, but you need to do it very, very carefully with all ethical and societal considerations. So with that, I close my presentation, and thank you very much for giving me this opportunity to present the report of the committee. Thank you very much.
Thank you. Thanks a lot, Professor Thelma, for very insightful and very substantive presentation of the report. I'm so thankful to you for bringing out it in form of your PowerPoint and also reflecting on the broad way forward that countries and societies have to keep a full view of things as the regulatory architecture. But more important than that, people's confidence in technology, I think, is something which is extremely essential. So thanks for a very substantive presentation. I would now request you to guide us through the session as you are also chairing the rest of it.
Okay, thank you very much. So it's now my pleasure to invite the first of the speakers. All of you would have seen the program. We have 3 topics. One, CRISPR and health. Second is CRISPR and agriculture. And the third, of course, is the oversight of the governance mechanisms. So we first have Dr. Devajyoti Chakravarti, who heads the RNA biology group at the CSIR Institute of Genomics and Integrative Biology, where he works as a senior scientist. He finished his molecular biology master's from West Bengal University and completed his PhD from Max Planck Institute of Molecular Cell Biology and Genetics at Dresden. And following a postdoctoral stint at the medical faculty in the Technical University Dresden, he came back to India, set up a lab, and he's doing some excellent work, which you will hear from him. And Devajyoti, you have 15 minutes to present your work. Please go ahead.
Can I? Yeah, so I think we can share the screen.
We can see you, not the screen yet. Yeah.
Is it possible now?
Not yet.
There's probably a small lag between the...
Yeah, I think that's what's happening. We just wait a couple of seconds. Yeah.
Still not there, or?
No. Sushma, you have enabled him, right? You have enabled him.
Yeah, I have shared it actually. It gave me the option to share, but I think even in your presentation, it took some time for it to appear.
Okay. Okay.
Small lag. It's still not there, right?
It's not there. Have you shared the slides with Sushma?
No, I haven't.
Okay,
shared.
because otherwise she could have. Oh, okay. I think it's just beginning. Yeah. It's beginning. You can go full. You can go. full slide mode.
Okay.
Yeah, please do
You
that.
should do
Yeah.
it. Oh, so thank you, ma'am. It's a great honor to finally see you virtually. We have been trying to bring you to our CRISPR workshop. That hasn't worked out so far, but I'm keeping fingers crossed for in the future. So yeah, it's really a great occasion, to be invited for this for this session. Of course, CRISPR is it's not something new. It's been in the news ever since it came. I think no other molecular biology tool has attracted so much of attention over the last decade or so, as much as CRISPR-Cas9. There has been movies made, Netflix, and whatnot. And it's in reality, it when you have a technology that can almost make you feel like you are playing God because you're rewriting the code of life. That's what happens. So we already have heard about the Nobel Prize, and Emmanuel Charpentier and Jennifer Doudna are the people are the two amazing scientists who have been awarded the 2020 Nobel Prize in Chemistry. And ever since the CRISPR was discovered and it was popularized in various media, the debate about who would win the Nobel Prize has been very, very strong. Just as the debate about who owns CRISPR and the rights to CRISPR is something that is still being debated in the law courts. Similarly, who would win the Nobel Prize has been a matter of great debate 8 to 10 years, and people are saying that CRISPR has got a Nobel Prize pretty soon, within 8 years. But in reality, people who have been working in this field, some of us have been thinking that way back in 2014 and 15 that maybe we would see the Nobel Prize in this year, and so. But it's also very important to understand that like any other field, a field actually progresses because of monumental work which is done by giants in the field, and this hasn't happened only in 2012. So therefore the field of CRISPR is not just about two heroes, but there are several heroes, and it's very important for us to understand that that's how any kind of scientific development actually progresses, with years and years of basic research, with years of people who are silently working in the lab for very, very little outcomes. But even those tiny things that Mojica saw in 1993, where he first saw that bacteria has certain types of sequences which are completely of a different origin, not from their own genome, and he had a very hard time publishing this paper. We as scientists, oftentimes we have to we are judged by how high we can actually publish our papers. CRISPR, the field of CRISPR is actually a typical example of how that can push a field back by almost 20 years. The first CRISPR papers were so outrageous these landmark papers that the elite journals, Science, Nature, Cell, actually rejected them. They said this was not possible, and they had to publish it in some kind of a much lower ranking journal, and not many people were interested in. Now, when you look at it and on the hindsight, you could have thought that if just we take notice of big profile publications, if CRISPR was published in a Nature article, perhaps the field would have been progressed even 10 years back. So that tells a little bit about how the field goes, how we look at science, and sometimes we really need to maybe just step back and say, there could be a different way of looking at the same thing. So yes, the field of CRISPR has several heroes. A lot of them, have worked on different aspects of it, and they are continuously working on it at different translational levels. Also, how to go from the basic biology to the translational biology of CRISPR, and I think there are a lot of scientists today, so I won't have to go into details about where this has come from. But very briefly, it is it's a part of bacterial adaptive immunity. So the first scientists, they were very, very surprised to see that the bacteria has got a lot of foreign DNA within a CRISPR locus in the genome of the bacteria, and they were not able to identify where this DNA or how the hell this bacteria could actually have these kind of foreign DNA integrated into their locus. And it was over time that they learned that this was a type of immunity. It's kind of a cellular immunity against an invading bacteriophage, which is a virus which kind of tries to kill the bacteria. And the bacteria, in a moment of crisis, managed to take up a part of the viral DNA and catalog it in its CRISPR locus, as you can see over here in this image. And it's almost like a librarian who puts a sticker and a kind of a catalog that, okay, this particular sticker is for this particular virus, this particular sticker is for this particular virus. And so the next time that the same virus comes in, it uses a very complex machinery of a protein, or a few proteins, I would say, and an RNA from that same locus to actually go and recognize that and attack. It's like there is a mole in the system which tells the bacteria that this virus has attacked you previously. So yes, I have the immunity, so that when it attacks me again, I can actually launch a kind of a reciprocative attack, and therefore chop the viral DNA into pieces, and in the process it can of course eliminate the virus. And then there was this was actually the work that most of that kind of formed the basis of Doudna and Charpentier's work with CRISPR. And then there was, of course, this very, very bright and really a prodigious scientist at MIT, Feng Zhang, who came and showed that you can put all these two things together into an eukaryotic cell in a mammalian cell, and actually break DNA in the mammalian cell as well, or cause a double-stranded break. So to cut a long story short, how this works is that if you have a Cas protein like this, it works almost like a molecular scissor. And it uses this kind of a blue RNA, which we call as a guide RNA now, and it goes and binds specifically to double-stranded DNA, specifically because the specificity is dictated by the sequence of this RNA. And so you can put any sequence that you like and take this entire machinery to any part of the genome that you are interested in. And once it goes over there, it binds to double-stranded DNA. It cuts the DNA, 3 base pairs upstream of a specific sequence. If this is a Streptococcus pyogenes Cas9, which is the most commonly used Cas9. And then once the double-stranded DNA break is formed, the cell tries to repair it, because it's not a normal thing to happen that you have a DNA break and it is not repaired. So the cell tries to repair it. In the process, it sometimes does a good job, it sometimes does a bad job. If it does a bad job, then you have insertions and deletions, which is what we need to be careful about. And as Professor Thelma said, that this is something that's very deleterious for the cell. So if you imagine using CRISPR for therapy, you do not want this to happen a lot, because then you are actually in the process of going to correct the sickle cell anemia mutation, you are making a deletion in the p53 gene, and God knows what will happen then. On the other hand, if you actually control things and make sure that there is a small piece of donor DNA, which is a foreign DNA, which can actually has sequences here that are similar to the site where the double-stranded break is formed, then if a lot of things happen together in the right manner, you can actually introduce this specific donor DNA in that specific location, and that is known as a homology directed repair, where you are actually integrating a foreign DNA into a site. That's very powerful, because that means that you can actually change the DNA of an organism or a cell at a very precise point. And that's what is the real power of CRISPR in actually making heritable DNA changes. And I would not go into details of how it has progressed from 87, when people began to look at curd and saw that bacteria in the curd has got some kind of immunity against viruses, till 2020, where we have finished the first genetic of sickle cell anemia correction in a human being. So it's a tremendous pace of development, and now CRISPR is there everywhere, whether it is in gene therapy, whether you're making CAR T therapies to combat cancer, whether you're making specific biofuels, where you are making the properties of these organisms better, in the food industry, genetically edited crops, material sciences, genetic studies make knockouts of primates, mouse, etc., gene regulation, activating genes, repressing genes, tagging genes, RNA targeting, drug development, to try to see what is the effect of a drug on an organism. You have 20,000 genes which can be knocked out and seen in the presence or the absence of a drug, to see which is that gene which is actually responding to a particular drug. And of course, diagnostics, which I'm going to come to you in a little while. So these are all the different fields that have been working on. We were also in our very, very small way have been trying to contribute to this working on CRISPR. I'm going to show you two very, very small stories, which I'm going to rapidly go through for the sake of time. One of them is, of course, something that we completed first phase of, which is sickle cell anemia. I mean, it's a hot topic. Sickle cell anemia, as like I said, the US conducted the first CRISPR trial for the first patient, Victoria Gray, and who, as I today in the afternoon, I was hearing a talk by Jennifer Doudna, where I learned that she's basically sickle cell anemia free in a way. So she has been cured. It's an amazing success story how CRISPR can really translate into medical sciences. But effectively, what this means is that one has to correct this mutation, which is the genetic mutation causing sickle cell anemia. This work we have been doing from 2017 onwards as a part of the CSR sickle cell anemia mission, very generously monitored by the Prime Minister's office, and done with Dr. Shobik Maiti at CSR IGIB and a lot of collaborators all over the country. Sickle cell anemia, you can see that India is even redder than the US. So actually more than 50% of cases are from India. And it's a huge problem. There are belts of sickle cell anemia individuals in the tribal populations and so on. And to cut a long story short, we had been working extensively to find out a CRISPR system that can actually work very, very efficiently to correct this. And this study came out last year. It was highlighted in PNS as one of the main studies in that issue. But effectively, what we recognized or what we characterized was that this orthogonal Cas9 from a different bacteria called Francisella novicida, which a lot of people overlook because of its large size, was actually it has evolved to become a highly specific genome editor, and it doesn't actually do edit targets. One of the points that was raised in the agency's report, as Professor Thelma said, is that it's very important that off targets be eliminated if you even want to think of going into germline, because in addition to what you are supposed to edit, if you edit a lot of other things, that is dangerous. So of course, we are very excited about this protein. We have engineered this protein into better versions. We're filing patents on that to make sure that we can enter into the therapeutic domain here in India as well. The other thing that has happened over the last 2-3 just to complete that, we are right now at the stage where we are going into preclinical trials very soon to see whether in the next 3-4 years we would be able to have enough data to convince the regulators that a CRISPR trial for genome editing can be possible in India as well. The other thing that I said was something that has become 2017 onwards is called CRISPR Diagnostics, and this is something that we have been also heavily investing in. Of course, we are not the first ones. The first CRISPR diagnostic papers came from MIT and UC Berkeley, both from the labs of Zhang and Jennifer Doudna. And now there are multiple companies that have actually started on this front: Sherlock Biosciences, Mammoth Biosciences, both using Cas12, Cas13, and other proteins which generally cause some kind of a collateral cleavage activity, causing a reporter readout. And when we saw that, this Francisella novicida Cas9 is highly specific in genome editing properties, we immediately thought that we could actually turn it into a diagnostic device. And that's when Fenuda was born. For the Bengali audience, it doesn't require any kind of introduction, but there was a Sherlock, there was a detector, so it needed an Indian detective. And when you have two Bengalis in the team, that's what happens. So we gave it the name FN-Cas9 Editor-Linked Uniform Detection Assay. Principle is that it doesn't tolerate mismatches in the target, which makes it give a different readout if it's a point mutation versus if that mutation is not there. And the net effect is that you can see this readout in the form of an agarose gel, in the form of a capillary electrophoresis, a fluorescent readout, or even in the form of a lateral flow. That's what I'm going to talk about in the next couple of slides. And we were already trying this out before the pandemic, and Dr. Shobik was leading a team here in Raipur, in a village in Raipur, again a hub of sickle cell carriers, where this is how the test currently is done, using a solubility test, a very crude test. It doesn't really tell very accurately whether you have a mutation or not. And this is in a primary or a tertiary healthcare center, actually, where kids are waiting for the prick and the blood to be extracted. If you look, some of these very young kids, they're not very too thrilled at the prospect of going in the queue and then getting their thing, fingers pricked. So we came up with a solution where they can actually just simply spit it into a tube. The saliva would be collected, and Fenuda can be done just within a couple of hours or so, and you can know without the need for any sequencing approaches whether a mutation is present or not. So it's a mutation detecting pipeline basically. And then, of course, COVID struck and everything turned upside down. And we felt that we had an assay which could actually be repurposed for COVID-19. That's when Feluda for COVID-19 came about, where once you collect the specimen or the saliva, it's a single step RT-PCR, which is not the same as real-time PCR, but a reverse transcription PCR, where the RNA is converted into DNA, amplified using biotin labeled primers, and then combining paper strip chemistry with that of CRISPR-Cas9 biology, we can have a readout within an hour or so. It's economical, requires just a normal PCR machine and in the first phase, and not a RT-PCR machine, and it also can have a semi-quantitative mode of readout. And this is how the readout actually looks like. So this is a positive sample or a positive control, and a negative sample and negative control, and you can see this. This happens within a couple of minutes, basically. Once you dip it into the final Cas9 RNP complex, you have this control line which is present in every strip, and then followed by this line over here, which tells you if your sample is actually positive for COVID-19. And of course,
Sorry for interruption. You're kind of nearly 20 minutes, so if you can.
I have only a one slide, and then I'm done.
Perfect.
So of course, Feluda is there. It's now possible to go down to very low copies, as you can see over here, almost 100 copies to 10 copies. It is assisted by a smartphone application where you can take a picture, and you can it's machine learning trained to tell you what is the outcome. And of course, now, as you know, that it is to be rolled out in probably in the next week or in a week and a half so by Tata Medical and Diagnostics. India, of course, needs more testing, and hopefully this can also be. This is, of course, if not the first, one of the world's first CRISPR-based paper strip tests after the Sherlock that has come from MIT. And like I said, this is the last slide. Is that the cost is low, and we also have a version which is doesn't require a PCR machine anymore, and that's what we are working extensively on, to have a possibility of doing a home test, CRISPR home test, in the next few months. We are really working to try to get this out of Feluda 2.0, if I might say. And these are the challenges that CRISPR hold for us. I won't go into details because there are other people who would be talking exactly about these things. But I think ethics, safety, and delivery system. So, we have not touched about affordability. We do not want the CRISPR to be a technology of the rich, but to make it more accessible. What are the steps to go in that direction? And it requires a lot of brainstorming, like the session that we are having today, to come up with sustainable plans. So with that, I'd like to end. A lot of people involved, and we are very generously funded. If you have any questions, would be more than happy to take it.
Thank you very much, Devajyoti. I understand that we're going to have the questions or discussion at the end of the session after everybody presents. So I now invite Dr. MK Reddy, who is currently a group leader in the plant molecular biology lab at the ICGB in New Delhi, a PhD from University of Hyderabad, and he currently works on identification and isolation of differentially expressed genes under different abiotic stress conditions using cDNA subtraction techniques, and develop tools for gene discovery in order to investigate plant architecture and adaptation to abiotic stress. So, Dr. Reddy, please, and you have 15 minutes.
Actually, I didn't know I had to present the slides, so I didn't prepare for that. Okay. So I will just talk on this technology, what in the agriculture we are doing on this technology. The originally, can they make it? Can they enlarge my slide? Can you put me on the full screen?
You can share your
do you have slides, Professor Reddy? Are you saying that?
No, I don't have slides. I'll just start because I didn't know the thing I have to present. I am only panel
No
discussion.
issues. Please go ahead. Please go ahead.
Enlarge my picture there, full screen. You can see me in a full. Yeah, okay. I cannot see myself like that, so that's okay. Originally, the CRISPR-Cas toolbox is designed to make the targeted double-strand DNA break in the living cell. But now these tools were improvised such that one could introduce any genetic modification in the genome in addition to the random deletion addition of nucleotides. It is like using the prime editing, base editing, and also the exact deletion of the excision of the base or the other things like that. So many things are improved with this technology. Now with this improvised technology and all the know-how is in the public domain, even a simple molecular biology lab can effectively use this technology for this genome editing in this. Compared to my earlier speakers, in plant system, the ethics and the safety is not a much concern, and also the off targets are not much concerned. I will tell you why this is like this. Mostly in agriculture systems, we make the heritable genome editing so that we edit the cell line and make the full plant, because the plants are a totipotency to develop into the full-fledged plant. So from these plants, we can generate the transgenic free, all the tools can be segregated out, and any off targets we can self cross and eliminate all the off targets within a reasonable time. So no problem in the plant systems. Using this technology, I will show you some of my results which I am doing in the rice to improve the crop improvement in the system. Increasing yield is one of the major importance in the rice system. For example, in rice, there are so many genes with a lot of what you call as a cultivation, a domestication for a long time. Many negative genes are accumulated in the system, and because of that, the yield potential is decreased. So we use the genome editing technology to knock out those negative genes and get the functional gain from these things. So for example, in increasing yield, we have several genes with a negative agronomic, when they express, they have what you call a negatively regulating the agronomic performance. For example, we use a tiller branching gene, the TB gene, which will suppress the tiller number in the rice. By suppressing the tiller number, you have a less number of tillers and the yield will decrease. When you knock out this gene, the number of tillers increases tremendously. Generally, in the rice, we get a 10 to 15 tillers in the rice plant. But by knocking out this gene, we can get up to 34 tillers. Each tiller will give the spike and a seed with this. By increasing from 13 tillers to 34 tillers is a tremendous jump in the yield. So we have done this in the rice, and we have shown the results, very encouraging. And because I didn't put the slides, so I cannot show you here the things like this. And another thing is a gene called dense erect panicle. The panicle should be erect and dense in terms of their spikelets. Because of this character, when you knock out this gene, the spike will be very dense with a more number of grains in this compared to the loose panicle in the rice things. So because of this gene knockout, so we successfully developed this gene knockout, and with almost 60% of grains per spike increase in the knockout mutant compared to their non-transgenic, non-edited lines. So 16% increase in the grain number per spike is a very huge jump compared to that. And another thing is a cytokinin oxidase gene 2, which is expresses in the developing spike of the inflorescence of the rice. When this cytokinin oxidase gene expresses, it oxidizes the plant hormone called cytokinin and make the cytokinin inactive. So if the cytokinin is a very important plant hormones, Because of this, the cell division and cell elongation will increases. And if the cytokinin oxidase expresses more, it inactivates the cytokinin. The inflorescence size is very small. When you knock out this cytokinin oxidase gene, the functional cytokinin will be more in the developing inflorescence, and because of the cytokinin, more cell divisions, more cell elongations happen. It's a larger spike will happen with a more grains. Almost the double the number of grains will happen in the knocked out this CKX edited line compared to the its non-edited lines. So all these mutants are available in the Japonica germplasm, and in the Japonica and in the Japan and China, these mutants are extensively used in their breeding system. But these mutants are not available in Indica germplasm. But we know the only the information about the what the knockout mutants are in the rice genome. So using that information, we are successfully developed these knockout mutants in Indica germplasm, and showed the tremendous increase in the yield potential by knocking out these negatively regulating genes. Now these genes are available because the Japonica germplasm we never use in our Indian breeding system mostly. So we can using this technology to create the those mutants which gives the high yielding in the germplasm. Otherwise, these are not known, not identified in the Indian germplasm. Because of the knowledge available from the Japonica germplasm, we can create these mutants without without the actual mutant required or actual breeding involved. We can generate these mutants with a very less time. Because what are the all the three mutations I described here? I got the double allelic mutation in the first generation itself. It means within a 1 year, I got the mutants and also the Cas9 removed from those lines. So without any transgenic imprint, I have a edited lines ready to use it, like this kind of thing. This will help us what you call a reducing the breeding time and unnecessary breeding cycles, and generate the required mutation without any genetic drag or any linkage drag along with this in a natural breeding systems. So we can use this technology very usefully in this rice yield potential increasing things. Another thing is increasing the quality of the rice. So increasing the quality of rice is very important for the this purpose. Suppose I have done a low glycemic index rice. So in the rice system, the starch is composed of amylose and amylopectin. So both are the polymer of glucose only. Amylose is the what you call the unbranched polymer of the glucose, and the amylopectin is the branched polymer of the glucose. When the our enzymes digest the starch, so many branches are there, many glucoses are released at a time, and increasing the blood glucose level very quickly with a high glycemic index. If the amylose content is more, then we can release only the from one end to the 1,1 glucose each cycle, and release the sustained release of the glucose. So there is no spike in the blood glucose level. So by changing this amylose to amylopectin, amylopectin to amylose is very simple by removing the starch branching enzymes. So if you knock out those starch branching enzymes, you can get a non-branched polymer of the glucose, that is a amylose content is more. So it is a hard rice and very what you call a cooking quality and the eating quality is better in these things. In a similarly, the low phytic acid producing rice. The phytic acid is an anti-nutrient which is undigested by the human system, monogastric animals. Because the phytic acid has a very negatively charged and it chelates all the positive minerals, and the bioavailability of the minerals are very less because of this low phytic acid. So we can use this last enzymatic step in the phytic acid synthesis, knock out that gene, so that we can have a low phytic acid rice with a which can be used for a improve the bioavailability of the minerals in the rice grain. Because in our vegetative diet, the availability of minerals is not the problem. It is the bioavailability, because all the grains has a large amount of phytic acid, act as an anti-nutrient, which is chelating the minerals which are not available. Otherwise, we have enough minerals in our food grains. But the bioavailability is the problem. So we can use this CRISPR technology to knock out that phytic acid synthesis and improve the bioavailability of the nutrients. And also the next thing we are working on this is a disease resistance. In several diseases, bacterial and fungal diseases are infecting the rice things. The main thing which we are following is the knocking out the susceptible genes. When the biotrophic fungus or bacteria infects the things, first it invades into the plant system and makes the innate immunity of the plant suppressed by expressing some susceptible genes. These susceptible, when the bacteria or the fungus, any biotrophic pathogen invaded into the plant system, it secreted some effector molecules which will go and bind to the particular genes which are called as a susceptible genes. These susceptible genes, when overexpressed, they directly or indirectly suppress the innate immunity of the plant and allow the pathogen to establish themselves as a biotrophic and feed on the living cells. So when you knock out these negative genes, then the biotrophic fungus cannot establish because it cannot overexpress those genes to suppress the innate immunity system. Using these systems, we are developing the rice blast resistance in the rice and also bacterial blight disease in the rice. So by knocking out these susceptible genes. Another thing which we are working on this is a herbicide tolerance. The herbicide tolerance is we are following here is a target site modification. When you apply herbicide, the herbicide go and bind to the very important plant enzymes and inhibit that enzymes. So this enzyme and herbicide interactions are disrupted by a single amino acid change in the target protein. So suppose if I am using this glyphosate herbicide, it will target the EPSP synthase gene and blocks the aromatic amino acid biosynthesis pathway and kill the plant. If I change 2 amino acids in the EPSP synthase without losing its enzymatic function, but because of the change of 2 amino acids, now the glyphosate cannot interact with the EPSP synthase. The plants are resistant to the glyphosate, and the crop plants are, if you engineered for this, the crop is resistant to the glyphosate, and all the weeds are susceptible, and you can kill the weeds with a chemical herbicide. Like that, we have developed the triple herbicide resistance in the rice. One is a EPSP synthase for using a glyphosate as a herbicide, and the acetolactone synthase, which is involved in the branched chain amino acids, that is a target enzyme target for the herbicide sulfonylurea. So all these enzymes, a single amino acid substitution is affect the herbicide synthesis. These enzymes are essential to the plant. They are only single copy genes. If you knock out these genes, the plant will die. You have to replace one amino acid with these things. We are using the base editing, prime editing technology to replace this amino acid replacement using this technology. And we are also using this technology to develop the genetic male sterility, because there are nuclear male sterile genes are there which are regulated by temperature. At a restricted temperature, the pollen development is not there when you knock out this gene. So these genes, genetic male sterile, because the rice plant is a bisexual plant, both male and female things are expressing in the same flower. So we cannot cross these plants for a large production of a hybrid seed. So we have to make the one sex temporarily knockout. So we use the male sterile, what is the temperature regulated male sterile genes to knock out. So these rice seeds were grown in the restricted temperature. There they act as a female flowers, and you can cross with anything and to make the hybrid seed production. Generally, compared to the self-pollinated crops, the hybrid seed gives the at least 20 to 25% advantage of the seed, but the seed production cost is more, then it will nullify the things. That's why we are using this CRISPR technology to develop a temperature sensitive male sterile lines to cross with a any particular other genotypes to make the high hybrid things in the rice. So these are some of the things which we already developed, and also we are developing all the vectors for the Indian contest for my own use, developing the primer editing, base editing, and also the knock-in kind of things, which is the very difficult at present, but we are developing the vectors to knock in the things. What is the difficulty is we are using the delivering the genome editing tools through agrobacterium mediated transformation. This mediated transformation will go and integrate into the chromosome, and from there it will produce the Cas9 protein, so it will come out of the integrated place and go for available in the cell for editing. At the same time, the guide RNA will express and comes out, but the repair template is going integrated into the genome. It is not coming out to the site of editing. So to overcome this, we have used the viral vectors. What is that? Gemini virus vectors. In that, we remove the code protein and put the this repair template, which is also come out as a circle Gemini virus genome from the integrated place and available with a high copy number for a repair purpose. Most probably in near future, we can solve this problem, which is a more difficult at present. The replacing or the knock in the new insert into the double stranded break is difficult. Now in future, in near future, I am sure we will solve this problem. Thank you. I'm sorry for not putting my slides there properly to explain to you, because I didn't understand that I'm only in the discussion panel. So I thought I will only participate in the discussion, not to show the things. Thank you.
Thank you very much, Dr. Reddy, for that comprehensive overview of the work that you're doing. Now, to save on time, we will just move on immediately to Dr. Oli Mathur. So Dr. Srinivas will tell me whether we have a chance to go beyond. Otherwise, we might compromise a little bit on the discussion time. So while Dr. Oli Mathur puts the slide on.
We will have time for discussion. We will have time.
Okay, perfect. Then it's perfect. Okay.
We go till 5:45 or 6, we can go.
Okay, so we will quickly go ahead now and hear Oli tell us. She is the head of the ICMR Bioethics Unit at the National Center for Disease Informatics and Research. She is a globally recognized expert in bioethics and has worked on several related projects and developing guidelines and regulations, especially for the emerging technologies in biomedical research. The Bioethics Unit of ICMR is actually a WHO collaborating center for strengthening all the ethics in biomedical and health research. And she's extensively involved in capacity building in this very important area and looking at the aspects of integrity in research and also working with several institutions in this. So I request Roli to now present
Thank you.
her thoughts on bioethics in 15 minutes, please. Yeah.
Yes. Thank you very much. I hope you all can hear me loud and clear and my slides are visible.
Yeah.
Okay, thank you. A very good evening. It's such a pleasure to be
hear you and see your slides. Yes.
It's a great pleasure to be part of this very eminent panel of experts, to be sharing a few thoughts on the Nobel Prize for CRISPR recently awarded to the women scientists. And I learned that we should not call them women scientists, but scientists. But I think it's a big breakthrough for not only genetics, but also, an example globally, a moral booster for a lot of women scientists across the globe. So thank you, Dr. Sachin Chaturvedi and Dr. Ravi Srinivas for this opportunity. And thank you, Chair Dr. Thelma, for the introduction. So, my slide is not moving. Yeah, okay, great, sorry. So the Nobel citation alludes briefly to the fact that the power of CRISPR-Cas9 technology also raises serious ethical and societal issues. And this helps to draw our attention to an important fact that revolves around connecting science, technology with society, for being able to reap the full benefits for betterment of mankind. So the capability for making precise changes to the human genome raises all kinds of difficult questions about how far we should go ahead with it. So it's so important to connect science with society and to make it ethical, to make something really successful. So it's critically important that when we are dealing with newer technologies, which have an untapped potential right now. We don't even know it has an unprecedented potential for making improvements in human health as well as agriculture and other fields. So on one hand, these discoveries have opened up new avenues for science.
Your voice is breaking, so maybe you could speak a little closer to your mic.
Okay, sorry.
I said you could speak a little closer into your mic. Your voice is breaking.
Okay. Sorry about that. I hope I'm better now. So it is critically important when dealing with newer technologies with an untapped potential for making improvements to human health. So as on one hand, these discoveries open new avenues for science, but on the other hand, they open up the need to connect, communicate, and engage as we learn the applications as well as the implications for the society. So with the scientific revolution that we have on hand, we have a host of probable benefits, but there may be some risks that may be seen. Now, it's very important that, sometimes the risks look very hypothetical at the moment. And it still remains to be seen how these risks will be higher or lower, because the technology has to be used in favor of the society in order to reap the benefits. So an important consideration that we all must think about is building public trust. And this cannot be built overnight or in 1 day or 2 days. It depends on how well we communicate, engage over and over. And therefore, as scientists, we have a responsibility to build this over a time and give some of our effort to this activity. Because public perception will depend, public perception of accepting the technology, accepting it as a, and how much effort we put in this exercise. There are a number of questions, of course. Often there is a disconnect between researchers and ethics. And often ethics is seen as a stumbling block. It delays research. There are a lot of discussions going on where, but if we look at the goal of the researchers, the ethics committees, the sponsors, the regulators, the government agencies, I think the goal of everyone is to provide the benefits, to improve the quality of research so that we have outcomes which can be used for public health benefit. So basically, we are all working on the same side and we need to work hand in hand on this. There are a number of questions about how the CRISPR would be put to, the benefits would be utilized, what uses can be permitted, how will people be able to access these, what frameworks do we need to regulate these, and also questions relating to ensuring equitable access to these powerful techniques. We don't want the powerful to get the technology, but the common man to not get it. So how do we really have to, because it should not lead to further dividing the gap between, people who can have or have not. So looking at the gene editing, I think you all understand the two types of gene editing that we heard from other speakers, somatic and germline. So what I wanted to tell you was that, the basic difference is just that these are heritable in case of germline. And therefore, there are some questions whether genome editing be used to avoid genetic diseases or can it be justified for genetic enhancement? Where do we put a line? What should be allowed? Who decides? How it is decided? Whether CRISPR germline should be allowed, for what conditions? What kind of heritable changes can be allowed to be inherited? So I think Dr. Thelma already mentioned a few in this regard. So looking at somatic cell gene editing, we find that a number of scientists have deliberated and feel that somatic cell gene editing requires very similar treatment to other research. It's not heritable change. And this the safety assessment or benefit risk assessment is therefore much easier. And one needs to ensure that there is no exploitation. It is carried out with appropriate counseling or consenting process, protecting the vulnerable persons. There may be some issues related to commercialization aspects because genetics is related to personalized medicine in case of human beings, and there may be some framework that we have. But when we look at the germline gene editing at the moment, we have many issues that we still have to resolve. A number of expert groups have deliberated and announced. It was in 2015, actually, that an international moratorium was announced, and then in 17 that there were some provisions being created about having follow-up and of intergenerational effects, which are quite unknown at the time. So basically, we are gradually moving forward. And of course, this was discussed, the scandal that came up 2 years ago with the birth of the twins who were gene edited for HIV. It was looked down upon by the whole world. Although if we look at it in terms of scientific advancement, it was a scientific development, but then it brought a lot of shame to the scientist who was actually imprisoned. So because he had faulted on many accounts, such as safety assessments, ethics review, inadequate informed consent, etc. And therefore, there is a need to have this governance framework which will serve the public interest, and need for widespread, broad and inclusive social debates to engage public, identify what kind of safeguards and monitoring can be put in place to develop these frameworks to help in the betterment of human health through right pathways. In India, if you look at the existing framework, and this is how it looks like, on your left, you see the ICMR National Ethical Guidelines 2017, which have very briefly spoken about the use of CRISPR technology and the safeguards that are needed under Section 10 in genetic testing and research. It may also be noted that now, as per new drugs and clinical trial rules, ICMR guidelines have been mandated to be followed for all biomedical health research in India. Last year, ICMR and DBT have jointly brought up this new guideline on gene therapy that you see. And these have discussed guidelines on product development, need for clinical trials for gene therapy, and provide a detailed description of requirement for research and clinical trials. The guidelines have also given a flow chart to explain the stepwise procedures, and they mention about the review committee on genetic modification of DBT and the institutional biosafety committee. These are some of the committees which are there involved in oversight mechanisms. And also, all clinical trials for product development needs to follow the new drugs and clinical trial rules. So as of now, the germline gene therapeutic or gene editing for therapeutic purposes in utero gene editing also is prohibited in India. And somatic is more socially acceptable, but then all clinical trial applications will need the approvals of various committees before being submitted to the Central Drug Standards and Control Organization, CDSCO, and be carried out as a clinical trial with a preclinical and clinical research module. So taking this forward, the issues are clear. What we need is a fair, honest, open discussion. A lot of efforts have been made by scientists in creating advocacy platforms and channels. Generally, scientists who develop these technologies in the lab, they publish their findings, but for meaningful outcomes, translation of these outcomes, these invaluable research findings, it's very important that these come out to the public. Therefore, there are efforts required in identifying all stakeholders, especially public, engaging with them inappropriately to remove any mistrust and to dispel fears. We should not build on negativity, but we should think about positivity and see how we can build more capacity to work on CRISPR. And as this is something new, and we will need to train scientists to do more research, which can be translated. So while doing research, the societal and human element should always be there in the design, and systems have to be created for appropriate counseling and monitoring follow-up, which will go a long way to increase knowledge base And infrastructure is required, good communication, discussions, engagement, building trust. And therefore, removing any unnecessary fear from the minds of the public is important. If required, further guidelines and regulatory framework also need to be strengthened for this. This is just a paper that was published 2 years ago. We had discussed the ethical considerations of using gene editing in the Indian context. And finally, coming to, looking at, this is a summary slide for me that, how to make technology acceptable and how to remove the unwarranted scare. And so, as we have discussed, we need the technology for betterment, and efforts have to go hand in hand to engage, educate, improve dialogue, understand societal concerns. And I think this webinar, as an example, is one important. We have to do many more and increase awareness, not only among public also, but also clinicians and researchers and regulators and agencies. Everybody needs to learn a lot more on this. And in terms of good quality research, we can bring out safe, affordable, accessible, reliable technology, which will be available to common man and once developed at a low cost. Publications are very important, whether they are positive publications or even negative findings need to be published. And these help to ensure accountability of stakeholders and protect public interest. And I would end with that. And it's important for India to invest in the ongoing debate to put in place appropriate ethical and regulatory framework. And I'd like to thank you all for your valuable time. Thank you.
Thank you very much, Rolly, for that overview from the Indian perspective, governance and the issues. So with this, I actually want to thank all the speakers for very effectively conveying. Devajyoti telling us what he does with the disease, a genetic disease, to how they actually contributed to developing a very rapid and an inexpensive tool for COVID testing, which is like the need of the hour. Dr. Reddy gave you sufficient number of examples of their own work on so many different traits that they were able to address using this technology. And Dr. does on what is the current status, the kind of debates which are already on between ICMR and DBT and other stakeholders, and the way forward. So we close the presentation per se, and I let Dr. Ravi Srinivas handle the question answer session.
Which actually now tell us that, we are on the verge of a major technological revolution through CRISPR. Now, there are a couple of questions which have come. The first question is on the health. Obviously, people are more worried about health as to what happens if these therapies or these gene therapies or whatever you call the genome editing therapies work out. So first question is to you, ma'am. The question is that it's a little complicated question, I know. Can there be an international consensus on that? Or it is possible that, there could be some rogue states or some rogue laboratories that will violate all guidelines and, run their own unethical practices? How do we ensure that there is something which could be controlled collectively by us?
So as I mentioned already during my presentation, we said that the decision to go ahead with this technology, especially the heritable human genome editing, lies with each country, provided they have all the guidelines and regulations in place. In case those are not in place, that's precisely where the international oversight mechanism, whether through the ISAP or through whatever will come from the WHO side, will ensure that such work cannot be done if the system is not in place, even if a country were permitting the use of HHGE for clinical use. So specifically to your first one, yes, I think if everybody appreciates the harm and the benefit which is brought about by this particular technology. And if you as a responsible society will take this forward, I believe that germline modification, there should be consensus, international consensus. Even if there's no international consensus, the second one, the global regulatory mechanisms will make sure that there cannot be rogue nations and rogue scientists who will encourage medical tourism of sorts. We have enough experience with assisted reproductive technology and a variety of other associated stuff. So it is our responsibility to go ahead and ensure that we work towards this, and which means every country has very serious debate and very serious regulatory mechanisms and oversight mechanisms in place together with international monitoring. Because science is very important. From what Rolly said, I didn't really see how deep the scientific aspects and their deliberations to keep track of the current progress is in our country. And therefore, I think we need to kind of go along with other international agencies and take, learn from them and actually work together.
Yeah, the second question, which is very interesting, of course, it is meant for all the panelists, because the questioner has asked, we are all talking of benefits, but are we really aware of the risks involved, of using this CRISPR or genome editing? And if so, how will scientists will tell us, how will they assess the risk? How will we know that which is good, which is risky, which is not risky? And then if there are going to be serious risks, then how will we be able to handle it? In the sense that, should we wait that something should happen, and then, or should we take some precautionary measures to ensure that these risks are handled even before? I know it's a complicated question, but I've condensed it because the question was long. So I think any one of the panelists can answer that. Question is, how do we know there are risks, and if so, how do we handle them?
Yeah. So, Devajyoti or Dr. Reddy, do you want to do that, or should I kind of start with?
Actually, in the plant system, we don't have a problem with this risk assessment, because any off-targeted systems can be eliminated by crossing out or selfing, segregating. All is possible in the plant system. That is not bothering. And also, if the agronomic performance is not there, these seeds are never be used in the competitive agriculture system. So they will be eliminated automatically.
And coming to the health sector, or to look at this technology from a biomedical point of view, this is where your institutional biosafety committee, your institutional ethical committee, your stem cell committee of the institution, there are multiple committees where you need to go through. Any investigator wanting to do this must have clearances from the institution to start with. And then, of course, you will have very detailed and very critical examination of the protocols, the question which the investigator is asking, and it will be looked at critically by these committees. That's number one. And therefore, I think that should be possible to do. And to talk about the risk assessment per se, if we are talking about embryos which are edited, you need to have a few of the cells from the early edited embryo to be actually used to look at off-target and actually the desired edit. You need to look at that, and which means you really ideally should be doing a complete genome sequencing, and the techniques are not in place. While it may be easy for you to identify the on-target, off-target could be at multiple places, and therefore a hypothesis-free approach has to be used, and we are not quite there. So assessing the risk, or even making sure that what you are doing is really only giving you the desired change, is the biggest challenge at this point. And that's where continuing research, even if it is using discarded embryos or embryos which are not being used for implantation, that research must go on to tell us how exactly the technology is going forward and what is our experience with those. Would you like to add something, Devajyoti?
No, I think mostly you've covered it nicely. Safety, efficacy is the priority, and like you clearly said, the technologies to measure the say in a hypothesis free manner where genomic off targets. I would also add that it's not just off target editing, but also DNA breaks and recombinations, which can happen due to the first generation CRISPR tools, which would rely on double strand break mediated editing. So I think the field is evolving also, and now we have no more the molecular scissors, but molecular pencils, as they say, for the editors and the prime editors, which do not require a DNA breaks. I think there's a lot of hope pinned on that because they promise to have lesser DNA breaks. But in the end, it is that the safety and the efficacy has to be very, very foolproof in order to make sure that there is a real a framework in place to how to go ahead after the basic biology is done to go into the trans.
Yeah, and just to add another point, that no amount of evidence coming from animal work will actually be a surrogate for what happens in human embryos. So it's got to be work on human embryos and those which are edited to be looked at before we can allow any of these or before we can assess risk.
Ma'am, if I may add a few examples, if you permit.
Yes,
Yes,
please.
Yes. So I would say there are a number of unclear risks of the technology at the moment, because as we understand, as scientists, you know more. But then as if you look from the public perception, then there are unclear risks, because there are a lot of hypothetical things and we have to do a lot of effort in removing those fears. For example, people have this thing about irreversible changes in germline, inaccurate gene editing, implications for future generations, interaction with other genetic variations or even environment, the high chances of being misused for prenatal testing, damaging further sex ratio, for example, or unmonitored and unreported fetal manipulations in the in vitro fertilization clinics across the country. Ethics of creating designer children with gene editing requires a lot of debate. Then, of course, the off-target mutations that we discussed, and the ethics of animal experimentation also comes into picture. These are just some of the examples, and therefore some effort is needed to prepare a paper on this to allay the fears. Thank you.
Yes. I actually see a question on congenital hearing loss.
The question is, how soon we will have CRISPR kits for home use? In the sense that just as we use measuring blood pressure or sugar testing, will we be able to have CRISPR kits so that we can use it home? This is a question which has come from one of the participants.
No, I think we are too far from that. I think we are too far from that. While something like the COVID testing kit that Dr. Devajyoti talked about, while that I'm sure they will make it possible that people could use it on their own. If you talk about anything else in terms of heritable human genome editing, then that's not going to be there. I'm almost sure. Because it involves a lot more infrastructure and expertise.
There is an interesting question which says, who will bear the responsibility, in the sense that in case of CRISPR risk, in the sense that a business invests for money in R&D, they develop a product, but then due to some regulatory or some aspects or something, it is not permitted or it is not, allowed for public use. So who will bear the risk? Should we have some mechanism where government also funds some sort of this risky thing which can actually result in some good benefit, but then very premature to tell in the beginning? So this is a question which I think Dr. Rowley can say, or any one of you can try to answer. Because the question says there are risks which the business may not be forthcoming if they are going to have regulations that may block.
Well, I don't think I've understood the question very fully, but from what I understood is that he's talking about if the government doesn't allow, then what happens to research and the outcomes. And I think the government is also putting in a lot of money to support good research on CRISPR. And there are grants available, whether it's DBT or DST or CSIR, there are grants available and researchers have to actually avail these grants for this. So I don't know whether I answered the question correctly, but that's my take on that. Yeah.
Yeah, I think, if I may add, what he's asking is also whether anybody will permit or if they don't permit. So the question is, if your question or if your project is something which has gone through with the respective committees, they have assessed the depth of the question that's being asked and also the methodology which is going to be used. And if it is for basic research, I think it's perfectly okay, provided it meets all the guidelines. The biggest issue or concern is, again, I say, only when you talk about heritable human genome editing, there are much major issues. Otherwise, all of the research which is currently funded, currently supported, are all because of permissions to do such work. So you already heard from Dr. Devajyoti what happens in a clinic, for a clinical condition or for a genetic disease. So you have to be able to put forth your project very clearly, and it is for the committee and the investigators to deliberate on it and go forward.
Yeah, that's nice. And here is an interesting question which actually talks about, which is a general question. What is the justice value of CRISPR? I mean, I do not understand the question, but the questioner wants to know what is the justice value of the CRISPR, in the sense that how justifiable will be CRISPR for different applications. Should we go for CRISPR in everything, or should we limit it to very specifics, or how do we think in terms of other alternative options? Because the question says, in some contexts, PGD can be a better option than CRISPR.
Yes.
So how do you assess these things?
Yeah. So when you talk about other options, for a CRISPR edited situation for an embryo is really, if it is the only method which is available to couples who cannot have genetically related children because of their mutation status, then a CRISPR has great value, provided it's ready to use. Otherwise, for a large number of diseases, we have several other options, including pre-implantation genetic testing for the mutation which is known in that particular family. And there is a way that you can screen out embryos which have the disease-causing mutation in 1 or 2 copies, depending on the condition. So there are all options. It's only for a very small number of situations where you would talk about a CRISPR-edited germline utilization in the clinical setup.
Yeah, here is a question which actually talks about something which maybe not all of us may be aware. Someone has asked us to, what is the question of freedom to operate in CRISPR, in the sense that who owns the CRISPR technology, and then how does it, who has the license to operate it, and are there intellectual property related issues in CRISPR that, Dr. Devajyoti talked about some cases. So who controls this technology, and where is the freedom to operate in this?
I think you should take it.
Yeah.
Yeah, so very pertinent question. So at the onset, let me tell you that Cas9 and anything that is obtained naturally cannot be patented. So there is no patent that is there on the Cas9 protein or the RNA components or anything that is used as a CRISPR complex. What is patented is the applications of CRISPR. And in that, there has been a huge fight over who owns the patent rights for using CRISPR for editing eukaryotic cells. So that is the controversial patent that I'm talking about. And at the moment, it seems that MIT holds the patent for that. There is a fight with the University of California, Berkeley. But if you go to the MIT website, you will have a very detailed discussion about the patent rights of CRISPR. If it is a non-human genome editing application or a non-eukaryotic genome editing application, then of course CRISPR is not having doesn't have any kind of a patent unless you make your own developments, you file your own patents, and there are many people around the world, including us, who have filed patents on CRISPR for different applications. But the key patent which everybody is concerned about as far as genome editing is owned by MIT, and it doesn't mean that it makes is that nobody can use CRISPR for genome editing. It only means that you have to enter into some kind of an agreement with the MIT legal team and probably pay license fees in order to use this technology for applications on areas which they are probably not working on. So there is also infringement of areas where they are working on, their startup companies are working on. So best would be if you are a company or a startup who wants to invest into this, if you have a legal help somewhere, to look into the patent portfolio. It's a very comprehensive, complex portfolio that we are, as academicians, are often sometimes not totally aware of the loopholes and what are the things. But yes, it is a very strongly guarded patent as far as eukaryotic genome editing is concerned, which is basically the crux of CRISPR to do the editing in human cells.
Quite interesting, because the other question is for Dr. Roli Mathur. The questioner says very simple thing: how will we communicate to the public these complex things? I mean, at what sort of level should we do it? Should we do it at the high school level? And then if the CRISPR, these technologies are very technical, public should understand not just technique, but also how will it affect them. So who will be doing that in India? The second part of the question is something like, is there any Indian perspective on ethics for CRISPR, or are we talking in the general global perspective, in the sense that what we talk of is what is being talked elsewhere? Is there any Indian ethics we can talk about CRISPR?
Okay. Thank you for the question. Yes, this is very much Indian perspective, and I did mention about the Indian guidelines and the regulations, and currently these topics are covered in the Indian guidelines, was what I mentioned. But like Dr. Thelma has also mentioned, this is an ever-evolving field, and we need to learn as the science is developing. With the global experience as to how international guidelines are developing, India will also keep revising its guidelines and keep coming up with the technology to put it to full use. Now, regarding community engagements or public engagement, this is the biggest challenge that as a scientist, we also need to feel responsible to be able to do it. And for that, what we need is communication in simple language. Now, webinars are one way of doing things to reach out to public. So, for example, when we do ICMR guidelines, when we do revisions, we do a number of public debates. We post the documents in simple manners on websites. We invite people to attend the meetings. Anybody from lay public is invited to be part of the discussion. Civil society is invited. So these discussions have to go along a long way. We must prepare the material in simple language, which is understandable for public, and widely write newspaper articles. So we have to engage with media. It could be social media or print media or the TV channels or others. So there are various ways of doing it, and a considerable amount of exercise is needed to really do a good communication, to have discussions at public forums. So this has to be done widely, and a lot of onus is on us to begin the discussions. I think it's high time in India that we start discussing this in a big way. Thank you.
Yes, thank you.
If I may add, two other groups of people who can contribute immensely to this are the clinicians and the counselors, because a counselor's job is to put it in the simplest language, as well as for a clinician. That's like a very effective way of getting it across to a lot of people who would want to kind of know more about it. whether because they're affected or otherwise. So that's something which is generally followed.
Okay, that's good. And I think this is the final question which has come. That question again, is something that talks about governance. This question, the questioner says, there is a commission which Dr. Thelma referred to, and then there is a WHO commission. But these two commissions, will they be sufficient? And after these commissions, what happens? Who will do the next steps? Will it be again left to the governments, or both NIS and WHO will once again come together and do something?
At the moment, at least the commission which brought out the report made it very clear that there has to be a continuing engagement and a revisit of all that has been taken stock of as of now. So there would be committees, international as well as national ones, which must be in place for all times to come, and there has to be a continuous evaluation and assessment of what is possible, what should be permitted, what is not to be permitted. It's not a one-time exercise, because technologies will develop, maybe improve tremendously, so there has to be stock taking from time to time, and that's the way both national and international efforts will go forward.
Perfect. So I think we will stop the question answer session now. But I'm glad to inform you that some of the attendees have expressed their interest in working on this particular communication. And then there are academics who have expressed their interest that they are willing to work together with the panelists on certain things, like somebody has said, I'm willing to work on embryological side, collaborate, I'm from this hospital. So I would request them to kindly contact the panelists individually and then coordinate with them as to whether you want to work with ICMR, or whether you want to work with IGIB, or whether you want to work with ICGEB. And one question which is again people have said is that we another academic institution has said we are also very keen to work with this area. So I request them again to contact the panelists whom they think are very relevant to their work. For if it's genetics, I think it should be Professor Thelma. So they can connect themselves directly with them and then see what they can do next. So we think with this I will end the question session. Now we will go for the vote of thanks. Actually, we wanted to end it by 5:30, but then we saw there are about 15 questions, so which I actually condensed into 6 or 7 so that we can answer together. Otherwise, questions I will go for another 45 minutes. So now I propose the vote of thanks. First of all, I would like to thank Professor Sachin Dwivedi for his excellent support and then for supporting all these initiatives. And then I would like to thank Professor Thelma for her ready to agree with this and then conducting this event very efficiently and for making a very excellent presentation. Madam, I actually have read your report and we are also familiar with the work on oil genomics. I should tell you that RIS has got a forum on Indian traditional medicine which works with Ayush on traditional medicine. So we will be, contacting you again in a different context. I thank Professor MK Reddy for agreeing to speak in this event at a very short notice. I thank Dr. Devajyoti Chakravarti to speak in this again at a very short notice. And I thank Dr. Roli Mathur, whom we know well, because we have been working with her on various ethical issues and she has been writing in our journal also for participating and then making a very excellent presentation. In this meeting, actually, I recognize the presence of Dr. Shruti Jandelwal, who is a postdoc with the Downda Laboratory. So she, Dr. Dev Sarkar, who was the former director of IASCR Mohali, informed us that she is interested in participating. So we said most welcome. So I recognize her presence and then we are glad that someone from that lab is attending this event. I thank the technical team at RIS, Sushma Bhatt Madam and then Mr. Soumya for making these things very efficiently. Then I thank my colleagues, Dr. Amit Kumar, Dr. Kapil Dhanraj and Dr. Sneha Sinha for ensuring that the event goes smoothly by interacting in social media and also by giving wide publicity, collating the questions and then putting them together. I thank all the panelists as well as those people who attended it for making this a very successful event. Let me tell you, this is a series of events which you are planning, and then this is the first of the event. The subsequent events will be much more focused. That there will be one webinar on governance, one could be on plant genomics, and then CRISPR, where we will focus more on the applications in India, because Varis has been working on GMOs and agriculture biotechnology for more than 3 decades now. So this is just a beginning. So please stay tuned. We will be interacting with you more, in a much more broader way, and then with also on very different aspects of CRISPR technology. Thank you once again. Thanks very much. Thank you.
Thank you very much.
Thank you all very much. Thank you.
Very much. It was a pleasure. Yes. Thank you very much.
Bye.
Yeah.